This is part 1 in the review for information for exam 3, and my plan is to break this into a few parts. Part 1 will cover viruses, and then in the next section I'll move on to eukaryotic pathogens. And my goal for this section is go over the introductory information, cover the basics, and then I'll spend time going into the viruses like influenza and HIV and maybe a couple others that we spent some time going into detail on, but I'm not going to go through the notes for every single virus because some of them, the information was fairly limited to what's just on the slides, and I'm not going to take the time to just read that back. So to start off with, just a reminder about why the organization of the class is set up the way it is. We're talking about viruses in lecture right now, but we're really not doing anything with viruses in lab. And we mentioned a couple reasons why that is. One is they're just flat out too small to see. So for bacteria, we can put them under the scope, but you even have to get to the 100x lens, put the oil on, get as close as you can just to see bacteria as tiny little specks. For comparison, this is to scale. If this is one E. coli cell, this is the size of most viruses that we're dealing with. They're really, really incredibly small, and some of them even smaller than that yet. This is a 75,000x magnification of rabies viruses, these tiny little particles right there. And so problem one is they're just so small, they're incredibly hard to study. Problem two is, and we mentioned this way back in exam 1, viruses are dependent on being inside of another living cell to replicate. So you can't really culture them in a medium like we can the bacteria the way we do in lab. So if you want to culture viruses, you're going to have to use living cell culture. For example, we would have to culture human tissue inside of a dish, which isn't that hard to do, but then you would have to infect those cells with the virus. That gets very dangerous because if you can infect the human cells in your lab culture with the virus, they can potentially infect you as well. And so it's a very risky thing to do. All right. And this should also be reviewed from exam 1, but I'll expand on it a little bit. So a virus is made up of, depending on whether it has an envelope or not, 2 or 3 parts. At its bare minimum, a virus is just capsid, which is sort of a rigid geometrical protein structure, and then at the center of that is a nucleic acid, either RNA or DNA. And I said before that this is sort of a strange thing because the nucleic acids, the RNA or DNA, its entire purpose is really just to give instructions for how to make more viruses. And so I've often said a virus is just really a set of instructions, the DNA or RNA, that code for making more containers to hold that virus, the capsid. And other than capsid protein, the other things that those nucleic acids, the RNA and DNA, code for are often enzymes like polymerases. We'll get to integrases in just a minute. But the polymerase part of this is kind of important. Since viruses can be either DNA or RNA based, things get a little bit tricky. And we mentioned these in class. A DNA based virus is pretty straightforward. Right? The standard sequence for living cells is DNA, RNA, protein. This is really straightforward because all living things, not viruses, but all living organisms have an enzyme, RNA polymerase, that can do this. If this is a DNA-based virus, it's also going to have to copy its DNA into more DNA so you can make more viruses, right? You've got to do the RNA thing so you can make protein because the capsid is made. All right, so that's pretty straightforward. On the other hand, if you are a virus that's made out of RNA, things get a little bit more tricky. And so if your primary genome is made out of RNA, the first part of this is easy. You go from RNA to protein, you end up making capsid proteins the same way that this DNA virus did up here. RNA to protein is just translation, uses a ribosome. Obviously, viruses don't have ribosomes, but they can use the host cells on ribosomes to make it happen. The problem is, in order to copy more viruses, you also have to make more copies of the RNA genome itself. You can't just make protein. And so somehow, this cell has to get from RNA to more RNA. That requires an enzyme that is different than normal RNA polymerase. It's called RNA-dependent RNA polymerase. And all it means is, normal RNA polymerase up here reads DNA and makes a copy of it in RNA form. This, same idea, only it's reading RNA and making more RNA. It's a direct copy. The thing is, it's unique to viruses. Bacteria don't have it. Humans don't have it. No living cell that's ever been discovered has the ability to do that because they don't really have a reason to. We copy our DNA to pass on genetic information. An RNA-based virus has to copy its RNA. The reason this is important is, since this is an enzyme that's specific to the virus, it makes a really nice drug target, or a good potential drug target. And I'll give you guys an example later of a hepatitis C medication that works very well by targeting that enzyme. Okay. The other way that this can happen, there's a sort of a third option. Clear some space here. Some RNA-based viruses, they're called retroviruses, which I've got a slide on later in the notes, they will actually copy their RNA backward into DNA using an enzyme called reverse transcriptase. Reverse transcriptase for reverse transcription. Normal transcription goes DNA to RNA. This is running backward, RNA to DNA. Another good example of a virus-specific enzyme, you don't find it in any other living cells, including humans. And so again, it makes a good drug target, and we'll get to that in HIV. So a retrovirus, same thing, RNA-based, so it can just go RNA to protein to make its capsid. It reverse transcribes RNA into DNA, and now, once it has that DNA, you can just go back into the normal flow of DNA to RNA to protein, and everything works out just fine. All right, so we've got the bare minimum down. You've got to have a capsid. You've got to have some nucleic acids to serve as instructions for how to make more viruses. The envelope is optional, but the vast majority of human viruses that infect humans do have an envelope. The reason for that is they give the virus an advantage. Now, viruses can't make their own envelopes made out of lipids, and obviously viruses don't have a metabolism, and so they can't produce fats on their own. So they essentially steal their envelope from the host cell membrane whenever they leave that host cell. And the advantage of that is taking a chunk of host cell membrane when they leave makes it easier for that virus to get into the next host cell that it's going to infect. And so it's almost like a bit of a cloaking device to disguise the virus. If it's covered in host cell membrane, it makes it less likely to be detected as something foreign. Okay. That said, even if you have an envelope, or this one down here is showing a non-enveloped virus, either way, if you're going to get inside the host cell, the thing that's required is docking to a receptor. So host cells don't just see anything with the membrane and automatically take them up into their cytoplasm. You have to have something to actually sort of trick the host cell into accepting the virus. And that something always means a surface protein on the envelope, often they're called envelope spikes. Text got cut off there, but you can see it. Binding to a receptor on the host cell. Right? This is the host cell down here. And the important part of this is it's specific. Right? This specific virus binds to a receptor on this specific host cell and triggers, some sort of endocytosis or membrane fusion, and the virus is going to be taken up inside. Obviously, that's very important for the virus because they have to be intracellular they have to have access to all the metabolic machinery of the host cell so they can replicate. And those two options are illustrated here. And it's pretty straightforward. Option one is you can actually just engulf the whole virus. That's essentially endocytosis, the way that you guys learned it. And that's shown here. Very straightforward. Virus attaches to host cell membrane. Host cells endocytose things based on the activation of receptors. And so if the virus can activate a receptor that triggers endocytosis in the host cell, right, it'll be taken in across the membrane. At some point, that membrane and the capsid have to be broken apart and break down to release the free DNA, and I'll write in here, or RNA if it's an RNA virus, so that DNA or RNA can start being read to replicate the virus. The other option is, and this only works for enveloped viruses, is something called membrane fusion, and it's similar except instead of having to trigger endocytosis, which isn't always easy for the virus, instead the two membranes, since the virus has an envelope, the membranes can fuse, sort of merge with each other, and then the virus's envelope sort of becomes a continuous part of the membrane. The capsid and the nucleic acid are then sort of injected or spit into the inside of the cell where the same thing can happen. Uncoat the capsid, free RNA or DNA, and you can move on there to start replicating more viruses. Okay. In this slide, I want to elaborate on this concept of host cell specificity. The specificity in this case means that in almost all cases, one specific virus infects one specific host cell type. And that doesn't mean one species like humans or birds or pigs or whatever. It means one cell type. So a virus infects hepatocytes in your liver, or it infects muscle fibers, or it infects epithelial cells in the kidney, or small intestine epithelial cells. Pick one. Very, very few viruses infect multiple cell types. Right. The reason for this, we've already laid out part of it. The virus has very specific spikes or surface proteins on its envelope or its capsid, and those proteins bind to very specific receptors on the host cell. The important part of this that often gets left out is the reason it's so specific is human cells, so the host cells inside of a person, don't have the same receptors. And so you'll often have a receptor that's unique to white blood cells, a specific subtype even of white blood cells. We'll use the example later of CD4 receptor with HIV. Only helper T cells have large amounts of that receptor, and that's why they're the only cell type in the body that's strongly affected by HIV. And so virus has its unique protein, host cell, very specific host cell type has its specific receptor, and that makes a match. This is one of the reasons, the main reason really, why viruses are so much more specialized than bacteria. They can't live in a fluid, they have to get inside a cell, and if you're going to get inside a cell, you have to manipulate the biology of that cell. Okay, once the virus is actually inside the cell, there's some more work to be done. And I'm going to quickly run through these steps. This is a sort of a very generalized viral life cycle. We've already covered part of this. Adsorption, that's just sticking to the host cell, and that's the receptor bit that we just talked about. Penetration and uncoating, we've been through that. That's the endocytosis or membrane fusion to get inside. The uncoating means you've got to take the viral capsid off. In most cases, it just becomes unstable inside the cell and it'll fall apart. That's a good thing for the virus because it needs to get a copy of its nucleic acids free anyway. So we've essentially covered those. The synthesis part of this is what I went through back earlier in the video where I talked about RNA versus DNA viruses, the different polymerases they use. The point is, you've got to make two things. You're going to have to make new protein for sure, Capsomeres are just capsid proteins and spikes. That's got to happen. You've got to make more nucleic acids. This illustration is using RNA, but the same thing would apply for DNA. Some of this, the synthesis part, is going to be done by viral enzymes like reverse transcriptase or that RNA-dependent RNA polymerase. Some of it's going to be done by host cell machinery. Some viruses use the host cell's DNA polymerase. They always use their ribosomes because viruses don't code for ribosomes. And you'll start putting together these individual pieces. You've got some capsid proteins, some spikes, some new nucleic acids, and they've got to be packaged together. The assembly part of this is mostly random, and I'll jump really quickly to this slide. I played this video a while back in class, and I'm not going to really play it again here, but you remember the idea. Viral capsid proteins are structured, their shape and charge is such that if they rattle around long enough, that they'll self-assemble into a geometrical structure. And the same thing's happening here. If you get enough capsid proteins and enough nucleic acids, the success rate isn't 100%, but there are so many viral pieces floating around that a large number of them will self-assemble, spontaneously assemble into a new virus. That new virus then has to be released or escaped. Sometimes this means the host cell stays alive and the viruses just sort of exit one at a time, taking some new membrane with them as they go. Sometimes that means the virus replicates to a very large number of new viruses until the host cell dies and the whole thing just ruptures out. Depends on the specific virus. All right, next thing I want to discuss are retroviruses, and I'll use, this is sort of a generalized discussion of retroviruses. We'll talk specifically about HIV, which is actually the virus we'll spend the most time talking about, so this is a good introduction to that. I already went over earlier the problem here. Retroviruses need a way to copy RNA into more RNA. They could use RNA-dependent RNA polymerase, but they don't have that gene. They evolved a different mechanism. Like I said, that mechanism is the viral RNA is copied into DNA by this enzyme, reverse transcriptase. We've already been through that. The goal is you need to have a way to get RNA into more RNA. Once you have DNA, you can go through normal transcription and translation. That's not a big deal. The other thing that I haven't mentioned yet that makes retroviruses unique and really terrible as far as their effect on human health is an enzyme called integrase. And what integrase does is takes that viral DNA, this molecule here, and that integrase enzyme latches onto the viral DNA, exports it, or I guess imports it into the nucleus of the host cell, and can actually splice that viral DNA. So this light blue chunk is showing you the viral DNA. This dark blue over here is the host cell DNA. And what the illustration is showing you is this enzyme has taken the virus's DNA, inserted it into the host cell's chromosome, the human chromosome, and made it permanent. And, what do human cells do with the DNA in their chromosomes? They transcribe it into RNA. They translate that RNA into protein. Translation isn't shown here, but you can assume it. And that protein plus that RNA makes a new virus. And so it's a really good strategy for the virus, and it's a really terrible thing for anyone infected with a retrovirus, because what it means is the virus is making permanent copies in the human genome of instructions for how to make more viruses. So even if your immune system got rid of every single HIV viral particle inside the entire body, you wouldn't be rid of the virus, because transcription and translation of more viral proteins is going to be happening all the time with that permanent copy there. And it's, at this point in time at least, it's impossible to get rid of. There's no way around it. And so the advantage of this system for the virus is it's making itself permanent. It's almost impossible to get rid of at that point. And your immune system can't cut chunks of DNA out of your own chromosomes. And so it's it's a devastating strategy as far as the perspective of the patient goes. The other problem is, the other real big downside for the host, the person, is you're having your chromosomes chunked full of viral DNA, and the location is completely random. But the problem is, if you get very unlucky, you can interrupt genes that matter. The best case scenario in here, if you put a piece of viral DNA inside of a human chromosome, three things can happen. Thing one is nothing. You might interrupt an area of DNA that's not very important, and nothing bad happens to the cell except that it starts making HIV. The other thing that can happen is you interrupt a gene that's so important that the cell just dies. That's actually the best case scenario, because you can just go through cell division and replace it with another cell. Probably the worst thing that can happen is you can interrupt genes that are oncogenic, meaning likely to cause cancer. And again, this is completely random, but if you insert enough pieces of DNA in enough different chromosomes of enough different cells, eventually it's going to happen. This is a summary of cancer cases that are attributed to infectious disease. And HIV kind of sadly doesn't cause a lot of cancer, partly because of the cell type that it infects, but mostly because patients don't live long enough to really have that happen in most parts of the world anyway. But some you recognize here, like hepatitis B and C with liver cancer, HPV with cervical cancer, some others that we'll get to later on in the lecture. Okay, beyond just cancer, viruses can cause other damaging effects to cells, and I'll leave this fairly summary, but the main point is all that viral, all those viral particles accumulating inside of a cell, just massive amounts of protein, unwanted DNA, RNA, it does strange things to a cell. One thing is sometimes big clusters of unassembled viral particles can just build up inside of a cell. You'll call those, this is a stain for inclusion bodies. They essentially start aggregating into big hunks of debris. The other thing that can happen is the large amounts of virus can really interfere with normal cell function, especially the cell division part of it. And so cells divide, but all those viruses make it difficult to assemble a mitotic spindle and really divide correctly. And so they start copying their chromosomes, but they don't really separate. And what you end up getting is these things called giant cells or syncytia. And this is showing you one. This is actually one enormous cell. Each one of these blue stained objects is one nucleus. And this is just 2 dimensions. In 3D, this thing probably has several dozen nuclei. It's enormous. This is a really common thing that happens. Okay, and lastly, this is just a very simple reminder. Viruses infect people. They also infect pretty much every other animal on the planet. They also infect bacteria. This is an EM of bacteriophage viruses infecting E. coli. And so just remember, viruses are opportunists that will take advantage of pretty much any cell they can possibly infect. This can have impacts on human health. Remember talking last section about E. coli picking up one of its virulence factors from a viral infection. And so oftentimes, viral infections of other cells can come around indirectly to infect humans in ways that you wouldn't quite expect. Okay, so at this point, this is leading us into specific viruses. Like I said, a lot of these I'm not going to spend a lot of time going into. I'll just give you maybe the main points. Herpes viruses, the big things that we focused on, one is latency, and I'll just take this opportunity to remind you why viruses are so much more often latent than bacteria. The main reason is viruses aren't alive. They don't have a metabolism. It's very, very easy for them to go dormant. Herpes viruses are an example of that. They can very easily lie dormant inside cells, most commonly nervous tissue, for months, years, even decades in the case of, for example, chickenpox to shingles. Take a very, very long time. They don't need to use any ATP. They're not really going through any cellular processes, and they can sit there for a very long time. And so as you're going through these, we looked at Herpes simplex viruses. We looked at varicella, Epstein-Barr. For each one of those, the symptoms and what they infect are laid out pretty clearly here. Just pay attention to the latency part, because it's on the exam. This is a good example. This, along with HIV, that I can use for the examples of latency. For the hepatitis viruses, I'll just give you a reminder. Make sure when you're studying, remember that hepatitis is a very broad term. All it means is liver inflammation. And so when you're going through hepatitis A, B, and C viruses, remember, they're really not very closely related. They're completely different viruses. They all just happen to infect liver tissue and cause inflammation. We spent maybe the most time talking about hepatitis C, and the one thing that I'll mention, because it is a little bit complicated, is the drug that we talked about. Since Hep C is an RNA virus, it's not a retrovirus, it has to use an RNA-dependent RNA polymerase. And for people that develop chronic Hep C infections, which is the majority, they're looking for a way to get rid of it. For a long time, there have been drugs that can sort of manage Hep C, mitigate symptoms, but not completely get rid of it. And we mentioned this drug. It's actually a prodrug, which is essentially an inhibitor to RNA-dependent RNA polymerase. And the thing that it actually does is it's basically fake RNA. And so the drug is essentially mimicking an RNA nucleotide. It's close enough that the enzyme, the RNA-dependent RNA polymerase, which I mentioned up here, will try to use it, but it's just off enough in structure that it doesn't actually work. And so it's basically just throwing a wrench in the gears of this enzyme. It's incredibly effective, working as a competitive inhibitor in that way. The cure rate is very high. The claims are as high as 90%. The problem is really expensive. And so the big fight now is who's going to pay for it, how is it going to be paid for. But from a pharmacology standpoint, it's great, very effective. And so if I give you a question on the exam about using medications to treat viruses, this is a great example. You're taking an enzyme, this guy, that's specific to viruses. You don't find it in humans. And so it's a nice target because it's viral specific, and it doesn't actually kill the virus, but it prevents infection or prevents replication well enough that it gives the immune system an advantage and can catch up and completely clear the virus. So at the end of this, for at least 90% of people, virus is completely gone. All right, these next few colds, RSV, norovirus, these are pretty straightforward. I mostly went with what's on the text off the slide. They fall into the category of viruses they're a real pain. They infect a lot of people, but the symptoms aren't so bad that they don't cause very many severe illnesses, very few deaths. And so take a look at them. They also all fall into the category of very high transmissibility. They're easily transmitted. They're just not very virulent. And so put that in context. We'll talk about those terms later on, and these are good examples of that. Influenza viruses, which orthomyxovirus is the family. Influenzas come in a variety of types, A, B, and C. We're not really going to differentiate. We're just going to talk about them generally. I did spend some time here going into the details of how flu virus changes rapidly from a genetic standpoint. And what I want you to see here is, you guys have a practical bit of knowledge about this. You know that a new influenza vaccine, a new influenza vaccine has to be made every year. The reason is two things. One is there are so many different strains of influenza that different strains sort of come around and cause problems from year to year. And so the strains that really infected a lot of people one year might not be the same ones that infect a lot of people the next year. The second thing is, even those strains that have been around for a long time, they go through small changes that make it difficult for the vaccine to continue being effective for a long time. And those changes are called antigenic drift and shift. And for some terminology, the term antigenic comes from antigen. An antigen is just something that your immune system recognizes to identify a virus or a bacterium, whatever it might be. In the case of influenza, those two antigens are hemagglutinin and neuroamidase, often just abbreviated to H and N. So when you hear H1N1, H3N2, those numbers are just different variants, different alleles of hemagglutinin and neuroamidase. And you can see them over here. There are capsid proteins, spike proteins on the virus. H antigen, hemagglutinin, is responsible for attachment. That's how influenza actually docks to your respiratory epithelial cells, because it is a respiratory infection. It got its name because it causes red blood cell agglutination. This really only happens in a lab. It doesn't really have a strong effect when it actually infects a person, so the name's not really significant for us. Neuroamidase is an enzyme that we're not going to go into a lot of details, but in order for the virus to escape its host cell, it needs that enzyme to cut a protein that it's attached to. And so don't worry about the details of that. Just know these are the two antigens that we use to name different strains of influenza. Okay. So how do they change? Antigenic drift and shift means the genes that code for H and N antigens, right? The genes inside, in this case, RNA. Influenza is an RNA virus. They change in two different ways. The easy one is drift, right? Viruses, bacteria, even humans and all other animals go through drift. That just means small mutations that build up over time. And so from year to year, really from day to day, and the life cycle of a virus, those genes will mutate, small mutation here, base pair there, and they accumulate over time. And so after months and years and decades of replicating, strains of influenza will start having different enough sequences that the H and N antigen will have slight mutations that make them different enough that your immune system can't detect them from memory if you were infected by that virus before. So that's drift. It's not unique to influenza. All viruses have it happen. The one that is unique is antigenic shift. And in order for shift to happen, influenza has something that is not completely unique, but it's the only one that we're going to talk about. Its genome has 8 pieces, or 8 segments, and that's what these 8 little coils are supposed to be showing inside of here. The idea is one of these pieces of DNA codes for H antigen. One of them codes for N antigen. And the idea is, if you have 2 different viruses, so let's say that this is H1 and N1, and I imagine a second virus here with all of its 8 RNA coils throughout it, and let's say that this is H2 and this is N3. What can happen, and this is incredibly rare, but when you have enormous numbers of viruses infecting billions of different people and animals for that matter, it'll happen eventually. Every once in a while, one individual person or animal gets infected by 2 viruses at the same time. And if these viruses are both replicating inside the same cell, it's possible that when their viral particles spill out and you package a new virus later on, instead of repackaging H1 and N1 together and H2 and N3 together, you'll get an arrangement that has, for example, H1 and N3, or it could be H2 and N1. You get the idea. And so if 2 different viruses infect the same cell at the same time, when they break apart and are replicated, the repackaging of their RNA segments can get mixed. That's antigenic shift. What you get then is a brand new combination. So instead of H1N1, H2N3, you could get H2N1, H1N3. This can happen in a lot of different formats. And when that new virus comes about, it doesn't necessarily mean that it's going to be bad, right? The new virus could be completely harmless. It could be not very different at all. It could also be really, really bad. It's just that we notice the bad ones. They make the news. And so this isn't a guarantee that you're going to get a very virulent virus, but it's definitely a possibility. All right, the last virus that I'm going to talk about in a lot of detail is HIV. And just a quick reminder, HIV is the virus, AIDS are the symptoms. It usually, even without treatment, takes years of having HIV to get to the point of being diagnosed with AIDS, and we'll get to the details of that later. The thing that makes HIV so virulent and also very transmissible is it's pretty new. It hasn't spent a lot of time evolving in humans. It is essentially a hybrid virus with chunks of sequence from two different simian immunodeficiency viruses. That's referring to viruses originating from a monkey. And the transfer to the humans, to humans, happened really only in the last 50 years. What that means is HIV really hasn't had enough time to be selected for not killing people. And that sounds kind of weird, but all it means is there's no advantage for HIV to be so deadly. But it hasn't really taken the time to evolve yet to not have that effect. And spend a little bit of time talking about how all this happens. The first thing is HIV, like all other viruses, is very specific to one cell type. That cell type is helper T cells, which is a type of lymphocyte, white blood cell. And it does this because the spikes on HIV specifically dock to a receptor complex, the main component of which is called CD4. There are other proteins that matter, but that's the big one. And the problem with this is, for people, helper T cells are very important in sort of coordinating the activity of the entire immune system. And so if you have a virus that's killing off your T cells, it makes it very, very difficult for your immune system to fend off any other infection. This is why, when someone actually succumbs to HIV, it's not really HIV that's killing them. It's not the immediate cause of death. It's usually something else like pneumonia, or that's the most common one. But lots of other infections that normally wouldn't be all that bad, sometimes even flu, that normally wouldn't be lethal, but when you don't have an immune system, those other secondary infections take over. They can't be fought off, and that's why CD4 cell count, how many CD4 positive cells does the patient have left, is one of the most common indicators of how effective HIV treatment is. And so just remember the specificity part. This is a good example of specific protein on the viral capsid binds to specific receptor on only one type of host cell, and that's where the helper T cell thing comes from. Now, the progress of what actually happens. The thing about HIV is it's latent, which means you have some initial symptoms when you're infected. It's often described as being similar to mono because both infect white blood cells. What this graph is showing you, this is the initial infection. The green line is showing you how much HIV is present in the patient. Those mono-like symptoms are coming from this first spike right here. Just like any other virus in a healthy person, when you get that spike of virus, your immune system will produce antibodies. Right? This orange line or yellow line is showing you antibody production. And so in the beginning, it works just fine. Viral levels go up, your immune system produces antibodies. Once you start producing antibodies, levels of the virus goes down because you're fighting it off. Everything works like it's supposed to. The problem is you can't completely get rid of the virus because it's hiding out inside those helper T cells. So unless you're going to just kill off all of your own T cells, it's hard to get rid of. The other thing, remember HIV is a retrovirus, it has integrase. And so even if you are really good at clearing out the viral particles, those helper T cells have HIV's genome integrated into their chromosomes. And so the level of virus, level of HIV, never goes to zero. It hangs out latently for years, 1, 2, 3, sometimes up to even 4 years without treatment for a really long time. While all that's happening, T cell count, right, so this is level of CD4 T cells, starts falling. And essentially, HIV is just holding out inside those T cells, knocking them off one at a time until T cell count starts falling further, further, further. Eventually, you get to a point where the immune system is really badly compromised from this. One of the definitions of AIDS is having a T cell count under 200 cells per microliter. There's nothing magic about that number, but it's about the place where things get bad enough that the patient starts to notice that they're sick all the time. They can't fight off other infections, and they start to feel weak. Often a really large loss of body weight accompanies it. Lots of secondary infections. Cancer can come up because of the integrase activity. At this point, you say they actually have AIDS, the acquired immunodeficiency syndrome. And at that point, unless they're treated very quickly, they're probably going to succumb to a secondary infection, because the paradox is the lower your T cell count gets, the worse the immune system is at fighting off the virus, and so viral particles go up. The more viruses you get, the more the T cell count drops, the more the T cell count drops, the more viruses there are, until obviously things eventually come crashing down at the end. So this latency issue is the big thing that I want to focus on with HIV. If HIV was just this, it would be like mono. Irritating, can be severe in some cases, very, very rarely actually causes death. The fact that it's latent makes it a whole different ballgame. And we talked about, one thing we mentioned in class is, why don't vaccines work? And the conclusion was, as of right now anyway, vaccines can't eliminate the latent period. When you take a vaccine, it primes your immune system to make antibodies ahead of time. And so instead of having to do this when you get infected, a vaccine will get your antibody levels high before you ever get exposed to HIV. What that would do is it would knock out this big peak, and so viral levels would never get high enough to cause those symptoms like mono at the beginning. But as of right now anyway, a vaccine can't get levels of the virus to zero. It just doesn't happen. And so even if you get them down very, very low, it's not zero, and that latent infection comes back. And so all a vaccine would really do is hide the initial symptoms, but the latency would eventually bring you back to where the patient was anyway, being diagnosed with AIDS. Okay. This stuff about distribution and frequency, I'm going to skip over. We did that in class, and it's pretty straightforward. I do want you to have at least some idea of drugs that are used to treat HIV, and usually they're given in a cocktail. We mentioned 3 of them. Fusion inhibitors that block the receptor that docks to CD4, reverse transcriptase inhibitors that I've already mentioned block reverse transcriptase so that the virus has a hard time copying its RNA back into DNA, and integrase inhibitors that block the integrase enzyme try to prevent it from incorporating the viral DNA into human chromosomes. The thing that I want you to see is that they all have in common, all 3 of these proteins, right, the surface receptor, the reverse transcriptase, and integrase, they're all viral specific. You don't find anything like this in a person, and so they make pretty good targets. And so because HIV is such a unique, strange virus, retroviruses are not incredibly common. The one upside is it has a pretty good set of drug targets. And so when someone's taking an HIV medication, what they're really taking is a big cocktail of all of these different inhibitors all together. And the effect is it works pretty well. You can't completely get rid of the virus, but you can slow it down significantly enough that a patient can live at least a mostly normal life and a close to normal life expectancy if they start treatment early enough. Okay, the last thing I'm going to do in this section, I'm not going to go through the whole bit on Ebola, because, again, we did that in class, and it's not really a big part of the exam. The part of it that I do want you to know well is we talked about this idea of transmissibility. And for the exam, I want you to be able to separate transmissibility from virulence. We spent a lot of time talking about virulence. What are the symptoms? How bad are they? What's the mortality rate? You could measure it in a lot of different ways. Transmissibility the concept is very straightforward. How much does it spread? And we talked a little bit about the details with, Ebola. I asked you to remember things like ID50. Transmissibility is sort of tied to that. How many viruses do you need to actually cause infection? And we put a number on it. That number, usually referred to as R naught, written as R sub 0, is just the average number of new cases that each case will cause. What that means is, if somebody's walking around with a viral infection, on average, how many new people will they infect? And the critical number is 1. If every person infects one more person, then the number of infected people, assuming people recover or die, the number of infected people will be stable. It'll stay the same. If the number is less than 1, then eventually the infection will sputter out and go away in the population. And if it's greater than 1, then it'll spread. And exactly how much greater than 1 tells you how fast it'll spread. So, a couple points. Viruses come, and bacteria too for that matter, come in all sorts of different combinations. You can have a very, very high R naught, but really weak virulence. These are things like colds, RSV, norovirus. They spread very, very quickly, but the symptoms really aren't all that bad. They're not terribly virulent. You can have a very low reproductive rate, or R naught, and a very strong virulence. This was my example of Ebola. Symptoms are awful. The virulence is just, about the worst that you can imagine. But it doesn't really spread all that quickly if you take some precautions. And so one of the main points that I talked about is virulence. The only way you can really manipulate virulence is with medication. So, for example, HIV meds make the virus a lot less virulent. This is effective. But a lot of viruses don't have effective medications. Transmissibility, or reproductive rate, you can have a really dramatic effect on, mostly just by hygiene. And so, in the case of Ebola, we talked about, simply being aware of high-risk areas, hand washing, not having contact with body fluids. This can take a very high reproductive number down to less than 1 very, very quickly. And so just because something is bad on the virulence scale doesn't mean that it has to cause an epidemic. And the problem with this is it's very hard to say, when there's a big reaction, maybe even overreaction to a virus like Ebola, it's impossible to say at the end, how much was it, okay, this wouldn't have been bad, we overreacted, it wasn't a big deal, and how much of it was, it would have been a big deal, but because we reacted so strongly, we got the transmissibility under control. And in the end, there's no way to really know. And so the whole point of this was, you can be aware of transmissibility and talk about it plainly without necessarily having to scare people terribly, which is what the rest of these notes are covering. So I'm going to leave it at that for this video, and in part 2, I'll pick up talking about eukaryotes.